So yeah, that's a very nice opportunity to just show my latest results. Let me just see if I can minimize this. And yeah, share with the audience and everybody at bioimaging what we've been doing the last 3 to 5 years. We're now at the point of putting together all these ideas. And we've been using many imaging modalities. We start with lifetime imaging, but we have also developed single molecule approaches. And now we are trying to integrate this into cryo-electron tomography. So yeah. Just let me introduce you the system, the biological system we are interested in, human immunodeficiency virus 1. And this is how it looks like. It's a retrovirus. And when it's actually popping out from the infected cell, it has this immature form. And you can actually see this characteristic mesh of the polyprotein called GAG all around the inner leaflet of the viral membrane, and then a few envelope glycoproteins that are also interrelated with the C-terminal domain, with the matrix and the GAG. And there is a protease, and right after egress, right after popping out from the infected cell, the protease starts its activity and actually ends up with this iconic capsid formation that contains the RNA. And this also has an impact on the envelope glycoprotein dynamics. And this was shown by Krauslich in 2012 with STED microscopy, that the sort of clustering of the envelope glycoprotein is more likely to happen in mature particles. So there is an interrelationship between the process of maturation and the dynamics of the envelope glycoprotein. And this is more in the context that I was talking about. So in here you have egress, incorporation of the envelope glycoprotein in immature particles, the process of maturation, and the impact of this process in envelope glycoprotein dynamics, and then how it engages in the next process of infection. There is also, from the structural perspective, something very interesting, and is that so far, with both crystallography and cryo-electron tomography, we have not resolved the so-called native ENV-closed structure. The envelope glycoprotein, I'll talk about this a little bit later, is a complex protein. It's a class 1 fusogen, and it's a dimer of trimers, so it's quite complex. But there is many structures out there in the PBD, but it was actually found with single molecule FRET that indeed the native state of primary isolates in its closed conformation is still not solved, is still not known. And this is actually really interesting, that perhaps it's related to this idea that we need to look at this process of maturation from an holistic point of view, not only sort of isolating the envelope glycoprotein from its from the whole process, from the virus itself. So this is the outline of my talk. I'm going to be telling you a little bit about what we have been doing in order to label the envelope glycoprotein in primary isolates. Also a little bit of single molecule imaging, and then how we came about the idea of coming up with a maturation biosensor, so that we can ascertain the particles that we're looking at, if they are mature, immature, or in the process of maturation. And in there is where we validated this approach with what we call functional correlative light and electron microscopy. And of course, we will end up with a few conclusions. And yeah, so this is, for this first chapter, this is the envelope glycoprotein. This is how it looks like. It's, again, a dimer of trimers, and each one of the subunits of the dimer is the so-called GP120 that contains the CD4 binding domain. And the GP41 is more like the fusion protein. And basically, if you look closer, there's a number of variable loops, and we have actually chosen the variable loop 4 to insert here a polypeptide domain that is going to actually be helping us then so that we can use nanobodies, single domain antibodies that will engage with this sort of polypeptidic insert that we're going to be using as a docking site. And we have chosen the V4 loop because there's reports out there showing that the V1, V2, and V3, they are glycosylated in a way that are quite important in the process of what we call tropism, because during the process of entry, HIV is going to actually interact with CD4, its main receptor, but also a couple of chemokine co-receptors. And this can be CCR5 or CXCR4. And there are some patients that over the course of the infection, after a couple of years, in order to avoid immunization by the production of broad neutralizing antibodies, they change this usage of the co-receptor, and they change what we say the tropism, and they go from using a CCR5 to a CXCR4. And therefore, we didn't want to interfere with this process. And we just seeked in the literature for a synthetic polypeptide that is going to be the docking site for nanobodies, and we found the AlphaTac, published in 2019 by Gotzke and co-workers. And this is what we thought it could be actually useful. So there is no other factors out there that could compete with the docking. And in order to understand beforehand where to insert this docking site, what we did is to use AlphaFold and to generate a number of different options. We started with 10, 20, 25, 26 options so that we could actually predict how the insertion of this polypeptide was actually impacting the overall structure, and also if the docking site was distorted or not. And indeed, in some occasions, AlphaFold was able to predict that inserting this polypeptide in the variable loop 4 was actually giving sort of a distorted version of the docking site that would not be actually functional, at least from the perspective of the docking of the single domain antibodies. Luckily, we found a couple that were actually predicted to be working. So then we decided to, try these ones and take a look at how much the docking of the nanobody is going to impact the overall structure with a clash analysis, but also functional assays of infectivity with the presence of different concentrations of the single domain antibody, which is quite a lot in comparison with the production of viruses, 5 nanomolar. We showed that there is no big impact as compared to the wild type, the insertion that we call the 415D2. These are actually numbers that are given by AlphaFold when you start playing with them. And this is from the infectivity point of view, but we also actually used fluorescence cross-correlation spectroscopy. And in here, we label the single domain antibodies with ATO 595 and 94. And then also we label the GAG between the capsid and the matrix with M turquoise. We will talk a little bit more about it later. We don't label the whole thing because it's been shown that labeling 100% the GAG is going to actually have a big impact on the process of maturation and infectivity. So we only label 10, 20, 40%, and then we actually add wild type GAG into the mix as well. But the main thing here for this particular experiment is that in fluorescence correlation and cross-correlation spectroscopy, we park the confocal beam and we allow particles to flow, to diffuse through this beam, and we recover a fluorescence trace. And then we can treat statistically these fluorescent traits to recover the so-called autocorrelation function. This autocorrelation function gives us an idea of the diffusion of the particles that are circulating around. And then if we do it in 2 colors, we can actually cross-correlate both autocorrelation functions. And this means that in this case, the nanobody together with the virus are co-diffusing together, and therefore there is engagement. And this is what we found when we see that the ones that AlphaFold predicted to be all right, we do see cross correlation, which is positive, and this is nice. So this means that there is a subpopulation of viruses that are engaging with the nanobody against the ones that AlphaFold considered to be not functional, in which the cross correlation is 0. And this is just the mature HIV particle without the docking site that also shows that, of course, there's no engagement. That's all sort of as a negative control. So we have now 2 layers of validation showing that, the infectivity seems to be the same when inserting this docking site from alpha-TAC into the variable loop 4 of the envelope glycoprotein. By the way, this is a primary isolate. This is quite important because there are many different envelope glycoproteins from HIV out there, but we want to use a strain that is as close to the clinic as possible, and therefore it keeps the so-called native state. So we want to investigate this unresolved closed state of the envelope glycoprotein. So the next thing we did, now that we have labeled the envelope glycoprotein and we have validated this labeling approach, is to start doing single molecule EMF imaging. And in here, what we actually did is to use a DNA paint, single molecule localization microscopy with DNA paint, originally developed by Jungmann. And basically, just as a reminder, we actually take the single domain antibodies, and in there is a docking strand with a DNA chain that is going to interact with the imager strand that contains the fluorophore. And in here, basically, the blinking comes from this sort of hybridation between the 2 DNA strands, the one that is floating around, the imager strand, that is going to engage for a few milliseconds with the docking strand. We can control the concentration, and we can actually improve quite well we can improve quite a lot the resolution as compared to other approaches, because we have tried in the past to use STORM, for instance, but the laser power and the signal to noise is not so good as compared to DNA paint. And this is also quite better as compared to STET. We can actually increase the resolution down to 5 nanometers as compared to STET that was actually giving around 30 nanometers in XY. The problem here is that, of course, when you produce viruses, you have a mixture of populations. These actually were more or less a few examples, but the problem here is that we need to know what we're looking at. When you produce a batch of viruses, you also produce exosomes that perhaps contain envelope glycoproteins, but they are not bona fide viruses. And among all these, there could be a small population of immature particles, mature particles partially cleaved, and this means that we need to really pinpoint which of these particles are real viruses from mature, immature, and exosomes. So in here, what we did is to establish the production of a biosensor. There's a number of biosensors to understand if single particles are mature or immature, but our version is that we are able to insert a fluorescent protein, in this case M turquoise 2, between the matrix and the capsid. And this can be done in proportions of 50-50 or in proportions of 30-70. And in here, as I said before, if you just produce a FRET couple like turquoise with YFP, for instance, and what will happen is that the big size of the fluorescent proteins, around 32 kilodaltons, is gonna impede the natural process of maturation, and is gonna sort of arrest the process of infection. So with our collaborator Arnaud Gauthier in Paris, we thought that the use of FAST, which is a really small scaffold of 10 kilodaltons, could be the solution. And basically what we do is to use FAST in the presence of a fluorogen. And this fluorogen can be a dark acceptor or a bright acceptor. And the idea here is that if you have the gag mesh of immature particles, there's going to be interaction by FRET between the donor, turquoise, and FAST in the presence of the fluorogen. And once the protease cleaves the whole process and the capsid starts forming, this thread profile is going to be disrupted. And the same idea can be done for many different colors. And we have actually shown that with lifetime imaging. We can do this with different donors from M-ametrin that turn out to be a really nice fluorescent proteins for lifetime. This is something that we want to study further, because one of the main things for establishing a good thread pair is that the donor lifetime is as close as possible as a single exponential. And it turned out that M-ametrin is really nice because you excite it at 405, but it has a long Stokes shift that emits in the green. And all of our experiments, not only in the context of single viruses, but also in the context of expressing it in cells, showed a really nice single exponential decay. Yeah, as I mentioned before, we've done this with M-turquoise too. We've done it with GFP. We've done it also in the orange with different scarlets, and in the far red and the red with MK2. And we've not only done this in the context of lifetime imaging, but also in the context of thread intensity-based approaches, especially, of course, not using dark acceptors, but bright acceptors, the small fluorogens. The advantage of having the fast between the matrix and the capsid is not only that it's a small entity, but also that it's a scaffold that can be used by many different fluorogens. So you can actually have as well all the spectra for the acceptor. Yeah, so we have developed a palette of fluorescent biosensors that can actually be used in different contexts. And this is also taking in mind that, for instance, when using particles, HIV particles in cells or in tissue, is going to be more attractive to have access to red and far red dyes that are also working. Okay, so the next round of characterization was to treat with mild concentrations of saquanivir, which is a protease inhibitor, our batches of viruses, and establish the analysis, the threat analysis by FLIM. And in here we see that there is at the beginning a very small population of immature particles that is modulated and increases linearly with the increasing concentration of the protease inhibitor. So everything seems to work fine. And what is also interesting is that this binary idea that there is mature and immature sort of conformations, perhaps is not true, that there's something in between. There's like partially cleaved viruses, and there is a third population that arises with this mild concentration of saquanivir. And yeah, we wanted to explore this a little bit more. But now we have a tool that will tell us which of the single particles are indeed mature, immature, and of course, if they don't express any of these fluorescent proteins, most likely they are actually exosomes, even if some of these exosomes could actually be decorated with the envelope glycoprotein that we're interested to look at. But yeah, we need to discard these events. And this is also to show with cryo-electron tomography that the insertion of these big fluorescent proteins, even if it's in a small 10–20% of the population of GAG, that altogether is around 2,000 units per virus, is not affecting the morphology of the immature and the mature particle. In here we have a nice capsid in its conical form. This is with GAG GFP doped with wild-type GAG. But in here is with the fast, and we also see that we have the conical capsid. This means that we have both hexamers and pentamers for the capsid, and in here we have the production of immature particles, the nice orientation and you know, this mesh of GAG in the inner viral leaflet that we were talking about before. So functional assays are also actually showing, and also structural assays, that the labeling that we are establishing is actually functional, is working relatively all right. So next, what we did is to really test if in cryo conditions, the biosensor was also functional. And the idea is that we're gonna vitrify the sample. We're gonna actually expose the cryo EM grids with our viruses. And here we see that the main challenge here is to get the right concentration that we can actually image with diffracted limited confocal microscopy equipped with FLIM. So this means that, of course, in 250 nanometers at the size of a pixel, we could have more than one particle, and this will only be seen later on in cryo. So the idea is to find the sweet spot for the concentration of viruses. And also, in cryo conditions, when doing lifetime imaging, we wanted to see what's going on with the fret phenomenon, and we wanted to see if the photon, once it's promoted to the lower unoccupied molecular orbital, is going to remain there longer. And this is really what happened. Now here with the phaser, we do see that the biosensor is also working in cryo conditions, which is very nice because in a way now, and this is why we call this functional, in a way now we can actually have a biosensor that will tell us beforehand which particles to look at, if there is full cleavage of the GAG, if it's partial, if it's a mature or immature particle. And in here we see that indeed, the addition of the fluorogen changes a little bit the fluorescence. And for mature enriched particles, we see a few that are immature, but the majority, of course, they have this long lifetime. When we actually produce with our protease inhibitor another batch and we add the acceptor, all of a sudden everything is quenched. So the biosensor is working nicely. And not only this, but in cryo conditions, the dynamic range is much bigger and the lifetimes are much longer, which is expected because of the stability of the sample at these low temperatures. And in here we continued and we did exactly what we said before. We have access to cryofilm. So we have a confocal imaging. We can actually select the particles based on the intensity and the lifetimes, and we can map them, and we can then just do cryo-electron tomography and really understand if the particles that we're looking at in 3 dimensions, they actually show nice conical capsids or perhaps tubular capsids or something in between. And this is what we can do. We can actually as well quantify the amount of particles and compare this with our biosensor that in our mature and immature batch, what is the percentage of immature particles, which turned out to be really small. The immature particles in our sort of mature batch is really small. And this is something that was seen before. It's something that if I come back, I can show you here that corresponds to a small number, but indeed we want to be sure. And this is something that is corroborated here. And when we use the protease inhibitor, it's working quite well. And the majority of particles are indeed mature particles with a big population of vesicles. And we can go further and we can classify the particles. And we are now showing that indeed this idea that there is sort of a mature and immature sort of a binary system is not right. There are different morphologies that are based on the equilibrium between the capsid that corresponds to pentamers and hexamers. And if this equilibrium is distorted, you get only hexamers, for instance, and you get this tubular shape. And if not, you can actually get a sort of an abnormal capsid formation. And if the equilibrium is right, you have then this conical shape. And this is something that can be informed in turn by the biosensor. So we are quite happy with that. And not only this, but now if we come back to our original single molecule imaging, we can as well determine which of the particles was actually an exosome. And just see that most of these exosomes, they incorporate a lot of the envelope glycoprotein, and this is because there is no restriction. This process is not regulated, but the processes where the GAC is present and the virus is produced, it is regulated, and therefore it presents less number of envelope glycoproteins. And also, this is something that was seen by Krauslich with STED. They tend to form these clusters, at least in mature particles. Also, we saw it for immature ones. But we can now be sure that what we see is a virus, and not only this, but thanks to the use of our biosensor, we can see if it's a virus that is functional and it contains a conical capsid. And then we can actually establish a holistic approach and really understand if in mature particles, the envelope glycoprotein tends to form these clusters, and if these clusters are at all functional. And this is how they look like when we actually separate mature from immature particles. In here, we have just a set of 12 different viruses, and we do see that they tend to form clusters indeed. And also, in the case of immature particles, they also form clusters, but you can actually see them more often as individual particles. Sort of the GAG might actually impede the diffusion of the envelope glycoprotein, and they tend to be a little bit more sparse. And we can as well, with the high resolution of DNA paint, we can actually determine the size of single envelope glycoproteins, and both in immature and mature particles. And this is quite interesting as well, because as I said at the beginning of my talk, the native state of primary isolates for envelope glycoproteins, they tend to be closed. And in the PBD, if you open one of the structures that have been solved by other groups like Peter Wong or Pamela Bjorkman, you will see that the envelope glycoprotein tends to be partially open. And the distances between V4 is much bigger than 12 nanometers. What we're measuring here with a resolution of 5 nanometers approximately is that, we have these distances of 7-8 nanometers that for the first time we're showing that this native state, is the natural state of the envelope glycoprotein. And this is perhaps the reason why rational vaccine design is not been successful so far. We don't have access to the structural identity of these closed conformations. And this is another view of how we can actually determine and collect individual envelope glycoproteins from real mature particles. We have also done a little bit of MinFlux, also combined with DNA paint in collaboration with Chris Tinnen in Harwell. And we can do that in 3D. The resolution here was isotropic as well, down to 5-4 nanometers, X, Y, and Z. And we're really happy because I think, we are now starting to image these single envelope glycoproteins in real viruses, in real replicative viruses. And I will finish my talk just with a number of conclusions. And this is just the efforts that we have put to really characterize this system, in which it seems that when going from mature to immature particles, we have this dynamic process that the EMF density changes. And there is no such thing as, there is a little bit of a continuous in the process of maturation, and the availability of the capsid as a pentamer and as an hexamer is quite important. This is something that was already shown by Leo James and other groups like Beijun Zhang using cryo-electron tomography, but it's something that it happens in real viruses, and there are a number of mutations that actually produce these sort of phenotypes, but it's interesting to see how they are actually impacting the process of envelope glycoprotein dynamics. And yeah, I'll finish with future perspectives. And what we want to do now is time-resolve threat dynamics, time-resolve the DNA paint, and of course, see what happens with envelope glycoproteins in both mature and immature particles when we add different ligands, not only the receptors and co-receptors, but also broadly neutralizing antibodies. We also want to induce the opening and closure of the envelope glycoprotein with mutants, but this is a little bit more tricky because every time you induce a small mutation to change a little bit the dynamics of opening and closure of the envelope glycoprotein, you destabilize the protein, and they might actually shed, and the petals of the envelope glycoprotein might be lost, you know. And we also want to take advantage that now all the system is really well characterized for DNA paint, so that we can apply RESI and really determine, this sort of two conformational states, the open and the closed one. And with this, I want to thank all the members of my lab, collaborators like Sabrina Simoncelli in charge of the DNA paint, Stuart, which is actually helping out with the mutants, Ray and Maud from the Rosalind Franklin Institute, helping out with nanobodies and the cryo-electron tomography. And with this, again, thanks to everyone for listening.